in Pajamas! Bum, bum, bum. Alright, you guys, welcome back. Today we're going to talk about identifying organelles in a plant cell. Now, you'll notice that a lot of these are similar to the animal cell, but there are going to be some differences. Alright, so without further ado, shall we get started? Let's start with the nucleus. So the nucleus is this entire structure here. So it's the entire thing. All of that is the nucleus. And we sometimes think of this as the control center for the cell, and that's simply because of what it holds inside of it, which is our genetic material, chromatin. Now, some of you might be like, Miss Komar, I thought that the DNA was a genetic material. You are correct. But the DNA can take different forms. One form we know of as chromosomes, which is when it's kind of all separated out into groups, and we see that during cell division. And another form that it can take is called chromatin. So this string is the chromatin. Now, what chromatin is, it's when the entire DNA sequence is all condensed down together within the nucleus. So the nucleus protects the chromatin. Also within the nucleus is this circular black structure, which is the nucleolus. Now, the purpose of the nucleolus is to make a particular organelle called ribosomes, which we'll get to in a little bit. Surrounding and protecting the nucleus, it has its own membrane. We can call this the nuclear membrane, the nuclear envelope, any one of those would be fine. And it protects the nucleus. Now, we want to be very, very careful about what enters the nucleus because we don't want anything to go in there and mess up our DNA. It's not going to give you superpowers. It could very likely give you death. That's the thing about biology, study of life, all living things die. But you don't want it to be because something messed with your DNA. Alright, now I know it's hard to see, but in the nuclear envelope, we have these little tiny circles, these little red circles. And those are the nuclear pores. I'm going to bring the computer a little closer so you can see them. So those red circles, like there, those are nuclear pores. They are openings inside of the nuclear envelope that allow certain things in and out. You do want to be able to allow some things in and out of the nucleus, but it's extremely, extremely specific what can go in and out, again, because you don't want things messing with your DNA. Alright. So we said that the nucleolus makes ribosomes. So let's start with that. You see all these little black dots everywhere? Those are ribosomes. So ribosomes are organelles that will help to make proteins. So we take our DNA, we copy it we copy sections of it in the form of mRNA. The mRNA leaves the nucleus, comes out here, gets picked up by a ribosome, and the ribosome allows a place where the mRNA and something called tRNA can get close enough together and line up properly so that we can put the amino acids in the correct order to make a protein. So a lot of that is done with ribosomes. They help to make protein. The nucleolus makes the ribosomes make the protein. And then once the protein is made, the ribosomes are going to take it over to this big structure here. Notice how it's covered in these dark dots? Because it's covered in ribosomes. We call it the rough ER. So the rough ER, or rough endoplasmic reticulum, is an organelle that modifies proteins. Once the ribosomes are done making proteins, we have an amino acid chain, but it's not a functional protein yet. It has to be folded up, sometimes added to other amino acid chains. needs to be tagged or modified in some way before it's considered a functional protein. And that's what the rough ER does. It is going to modify the proteins so that they are ready to go out and be working proteins. We do have something similar to the rough ER. You notice this is the same coloration, slightly different shape, and no ribosomes on it. That is the smooth ER. So what the smooth ER is, instead of modifying proteins, it's going to make lipids and steroids and cholesterol, things like that. There is good cholesterol that helps your body to function properly. And a lot of that is going to be made by the smooth ER. The smooth ER can store some proteins, but again, its primary function is to make lipids. And yes, lipids are fats, but that doesn't mean that they're bad. Remember, the membranes of cells, including any membranes within the cell in organelles, like the nuclear membrane, the membrane of the rough and smooth ER, the membrane of the mitochondria or the chloroplast, or any other organelle like the Golgi. All of those are made using lipids, and specifically, like with many of them, phospholipids. Well, that means that we have to be able to make lipids if we want to maintain all of these membranes. So that's going to be taken care of by our smooth ER. All right. So when a substance that was made by the cell, whether it's proteins or lipids, are all done and they need to be sent out of the cell, they get taken to the Golgi apparatus or Golgi body, Golgi complex. It's all the same thing. This is like the post office of the cell. And what that means is, let's say we made a particular antibody. Antibodies are proteins. So this cell made antibodies. The finished antibody is going to be sent over to the Golgi body. And there it's going to be tagged and packaged, meaning that it's going to receive these tags on it that help the rest of the body, the rest of the cells of that organism to recognize it as self, as opposed to foreign. This way, it won't get attacked by other antibodies or other parts of immune cells. So it gets tagged, and then they get packaged in these little vesicles, which get sent to the membrane, and it'll push and expel the finished product out of the cell. Now, this works in animal and plant cells. Yes, plants make hormones. They do. And they have to be sent out of the cell via the Golgi. They have to package it and send it out. All right, now the mighty mitochondria. So a lot of times kids call this the powerhouse of the cell, which I will admit torments me to no end. It's this guy right here. And we also have one right there. Because when kids tell me that's the powerhouse of the cell, that still doesn't tell me what does it do. And a lot of times when I ask kid, well, what is mitochondria? I'm looking for its function. So it's an organelle that makes energy. How does it do this? By taking glucose molecules and breaking the bonds. When it breaks the bonds of the glucose, it releases the energy stored within those bonds and transfers it to ATP. ATP is another molecule, but the thing about ATP is it can be sent out and every organelle in the cell can use it. It's an energy currency. So it has stored energy in it courtesy of the mitochondria taking the energy from the glucose bonds and storing it in the ATP. And then that can come over here to help power the Golgi apparatus as it's tagging molecules. All right, let's see. The chloroplasts. These are fun little guys. So we have chloroplasts here, 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 and right there. So what the chloroplast does is, well, it does photosynthesis. And that's a special process where the plant has taken in water and carbon dioxide, and in the presence of sunlight or some kind of energetic light, it begins a reaction to break apart the bonds of the water and the carbon dioxide and reform it into glucose. Now, the reason why plants have this and we don't is because I, as an animal, I get my glucose from what I eat. So I ingest it. This helps me to provide glucose for my cells so that the mitochondria can provide energy to keep my cells alive. But most plants, they can't really eat. So how do they get their glucose? They actually have to make it themselves via the chloroplast. All right. Let's see. We have the vacuole. Now you'll notice in plant cells, the vacuole tends to be a lot larger than it was in the animal cells. So it still does storage. It stores extra water, minerals, molecules, ions, etc. But it has another job. Its job is to also provide strength and support to the overall cell. So it does provide some rigidity in the cell to make it stronger. Trees can get pretty darn big. They don't have an internal skeleton like we do to support that. So they have a few other ways that they can strengthen their insides and allow for larger growth. One being a vacuole, and the other being the cell wall. So the cell wall is this green barrier on the outside. Now it's this thick, strong structure that's made up of a lot of cellulose and other materials. And again, it's to provide strength to the cell. It provides a whole extra barrier to protect what goes in and out. But more importantly, it keeps the cell strong so that you can have cells on top of cells on top of cells and allow for larger organism growth, such as trees or bushes or any kind of plant that can grow big. A lot of that is based and thanks to the cellulose in the cell wall, as well as the rigidity of the vacuole. Underneath the cell wall, we have this purple part. That is our cell membrane. So they still have a cell membrane. It's this purple layer in between the inside of the cell and the cell wall. And just like with animal cells, it's a selectively permeable barrier that will allow some things in and some things out of the cell, but only specific things in and out. It keeps the good stuff in and it tries to keep the bad stuff out. All right, let's see. We have our lysosomes, which are these blue circles, and our peroxisomes, which are these pink circles. Now, lysosomes and peroxisomes are both organelles that help to break down molecules and recycle them. So whether it's bacteria that got into the cell or viruses or an organelle that's no longer functional, anything that needs to get broken down, recycled as waste, that's taken care of by these two. But they do it in different ways. The peroxisomes are filled with hydrogen peroxide, and they use that hydrogen peroxide to break down and recycle materials. Lysosomes, though, they're filled with enzymes, and they use those enzymes to break down and recycle materials. All right, let's see. We have then all the white area in between. So all the white, that is going to be our cytoplasm. Cytoplasm is that jelly-like substance that fills up the cell. It supports the organelles, helps to kind of float them and move them around as needed, and just kind of protects the inside of the cell. And last but not least, I don't show them up here in the picture because it would get really, really busy, But throughout the cytoplasm, we also have these elements called cytoskeleton. And what the cytoskeleton does, just like how our internal bones support our structures and our organs, that's what the cytoskeleton does. It's these pieces that go all throughout the cytoplasm. It connects to the different organelles and helps to keep them in place, helps them to move through the cytoplasm as needed, and it just provides strength and structure within the cell. Let's see. The parts of the cytoskeleton include things like microfilaments, microtubules, and some intermediate filaments. There's a bunch of different ones. But yeah, I think that kind of sums it up. Alright, so hopefully this helped you to understand plant cells. And just, if you have questions, definitely let me know in class or through Google Classroom or email. But in the meantime, you guys, take care. Just be awesome and be the great people I know you are. In the meantime, stay safe and stay healthy.